Questions? No questions? Okay. There's a poll that you can fill out if you like. And then,
On that part
uh-huh.
that we did, I did one and submitted it, but it says it's been accepted, but it hasn't been graded.
Oh, okay.
I don't know if they get graded later or just start exclusively.
All of them will have to be changed because everyone's getting a 0 because of the way it's set up. And then I'm going to change that to 12.5 points. I thought I sent, did I not send an email out about that? Maybe I did, yeah. It's just the new system, the old system, you could actually have a poll count as a grade. So you could give, regardless of what your choice was, then you could say that's, you can do it. The polls in the new system, you can't do that. I'm sure there's probably some way of doing an exam that way, but I couldn't find one. And they were looking, so, as long as you took it by that time, then when you go in, you'll see your grade change when I get around to changing them. Okay.
When you can start posting quizzes online?
I'm sorry?
When you can start posting quizzes online?
We're going to, good question. What about quizzes? Okay. The quizzes you'll have roughly 2 quizzes before each exam, and they will be close to the exam day. So I tend to backload, so you will always have a quiz the day before the exam. And then you may have one a week before then. So they'll basically, it's, it's obvious, and they've of course done the studies, if you do them closer when you have the exam, you retain the knowledge. So quizzes you'll have, in class I'll let 20 minutes for the quiz, so that's how much time you'll have for the quiz, very short answer, multiple choice. And then the next poll that will go up will be, when do you want the first exam? And because I think if you look in the syllabus, I think February 1 is listed as sort of that rough time for the first exam. I'm sorry?
2, 3, water relations, chapter 1 and 2, the week of.
Okay, yeah, because it's just a rough, it's not, it's just a rough time. The exam is not set on any particular day. The day you do have an exam, there won't be class that day. Yes, ma'am.
I thought you said, are the quizzes going to be online?
Yes, quizzes are going to be online.
Are we going to like the lab or students are we taking them in class? Like 20 minutes?
No, I said in class I give 20 minutes, so I'll give you 20 minutes online to take the exam. I'm sorry. I didn't mean you don't have to come here. You can take it from wherever you take it.
But if you are, do you take it in class?
Well, no. I mean, unless you bring your computer and take it. I'm just saying the statement I made was when I gave the quizzes in class, you had 20 minutes to take it. So online you'll have 20 minutes to take it. Okay, so again, same amount of time. You'll have 50 minutes for the exams because that's the prescribed amount of time for in-class exams. Okay, so if you're taking the course online or you're taking it here, you're going to have the same exact time length, the same exact kinds of, or the same exact questions, the whole 9 yards. Okay, except the difference is, even if you come to class, then originally I was going to have the people that signed up for the class to come and take it from 10:20 to 11. And then I thought about that and talked to a couple of people and said, well, give you guys the same amount of leeway, so you may have a 12-hour window where you can go in and take it at any given 50 minutes in that 12-hour window. Okay? Because then again, if I made you come here, then see, the whole thing that I'm trying to do is make the online and the in-class as absolutely close as possible. And I don't even know if anybody that's taking it online is even not here. You know, so far the people I've interacted with on Google Plus are all here. They just had a conflict and they couldn't take it. You know, so the whole point is to make it as close as possible to you being here. Okay, so there's other philosophies where online you have to do, you have to engage students. Well, we'll do that, but if I give a review session where it's a Google Hangout on air, then you can sit in here and watch me talk, or you can be online and watch me talk and answer questions. Now, the first couple of times, no one's done this, okay? But I don't want to use the equipment that's built into WebCT is just a bitch to use. I mean, if you want to talk about a slide, I can't upload all the PowerPoints. I have to upload a slide. And so I've got to go find that PowerPoint, find that slide, and upload that slide. Okay? Google Hangouts, I can just start the slides from right in the middle of it. And you all know, most of you have Gmail, so you have a good knowledge of that. And Google Hangouts is a little bit different, you know. So so there might be some questions that are coming up the side of the screen, and one of you are going to have to say, hey, someone just asked a question, because, you know, I may, you know, but we'll get it done. And I'm going to have several sessions, so, yeah, and then you're going to be using this, you know. Google's obviously taken over the world, and using these kind of tools, you know, are going to become common. And I mean, you can buy GoToMeetings, which costs a lot of money, or you can do the same thing when you're inside Gmail. You can do Google Hangouts in the middle of Gmail, and everyone in here raised their hand up when I asked if you had a Gmail account. So it's trying to get as cheap as possible for all of us. And so these things become more, and so, you know, and also if you struggle around with finding out how to use Hangouts, okay, you've learned something. There's nothing wrong with that. Okay? Just if you do Google Hangouts and send me a message or something, or send MEPS 313 to me, that way I'll know you're in the class and I'll put you in the right group. And I think I know I sent an email out. If you send me an email, put MEPS 313 as one word in the subject, and then that will go into a subdirectory that I'll be sure and look at all the time. Because otherwise, it will go into one of 10 or 15 different subdirectories in my Gmail account. I don't know why your message would go here and another message would go there, but it happens and I don't see them then. So just put MEPS 313 in the subject line, and that'll go into this class subdirectory, which I'll see immediately. There's a message in there. Okay? Okay. So today we're going to talk about organelles. Oh, you had a question? Yes, ma'am.
So are we going to decide on the first session?
Yeah.
Is there going to be like a poll? Or?
There'll be a poll up. Yeah.
Okay.
Yep, there'll be a poll. Usually, if there's 3 or 4 dates, one date will be more, so we won't have like 33%, 33%. It'll be, one of them will be 50 or 60%, and the other 2 will be scattered. So, yeah, you'll be able to decide. Okay. So, today you're going to see a lot of different kinds of cartoons that are showing plant organelles. And so far we've talked about the cell wall, and then we talked about membranes. We actually talked about mostly the plasma membrane. And then now we're going to talk about what happens when cells take membranes and enclose things inside the cell, and then use even other membranes to form things called organelles. And again, I think I mentioned this previously. if there are two, well, there's a lot, but the main inside the cell, the main differences between an animal and a plant cell is the chloroplast, of course, in a plant cell, and this vacuole. And if this were a mature plant cell, this room, the vacuole would occupy almost all the space. And there'd be a little bit, maybe a foot of cytoplasm coming from the cell wall. And then the rest of it is just this immense vacuole. And so the vacuole is an organelle. It's called a tonoplast. And it has a lot of functions. We're going to talk about a few of them. Some other things that I mentioned earlier that I said we'd be talking again about are plasmodesmata. What are those? Anybody remember? Yeah. They're actually holes between cells that are actually membrane lines, so things can move from cell to cell without crossing the cell membrane. Okay, last time we talked about active transport and transport. So getting things into cells and between cells is a big deal. Okay, so now we have a mechanism where things can move through the cell wall through a hole. So there's no transport required. It's also a big deal, and that's why we think a lot of viruses are transmitted. The cytoskeleton we're only going to mention briefly, very complex. And rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi we'll talk about. Let me tell you now that if for some reason we were now to be able to first look at these cells, but we have the knowledge that we have, we might name the chloroplast a chloroplast, but everything else would be part of the intermembrane system. So when these things were first seen over 100 years ago, they were seen as distinct units. But these things work together. So the smooth, the rough ER, the nuclear membrane, the Golgi, the vesicles from the Golgi, that's all part of a system that moves material around the cell. So we give them names, but again, if we were to name them now, it would be the loading dock, the shipping department, because they're all part of moving stuff from the nucleus all the way out, if necessary, to the cell wall. Okay? Very complex. Let's look at an active cell. What's this organelle here? Can anybody tell me what that is? Can you see where I'm pointing to it? Anybody know? That's a mitochondria. Here's the nucleus, chromatin you may have heard of course. Here's the cell wall blown up a little bit. Here's another mitochondria. Here's the endoplasmic reticulum. What are these things? Anybody want to know? Anyone want to guess what those things are? What are they? What would you say? No, they're pretty obvious what those are. You got to have good imagination. Those are Golgi bodies. Okay? See, what you get in this class is not the classic one shot that you see the same Golgi in every plant phys textbook, which, by the way, was done by a guy here at A&M. Dr. Hilton Mollenhauer, who's retired now, was the first person to describe Golgi in plants. And he did a really, and you see that same one micrograph, he got that perfect micrograph, that perfect block. And when you section these things, anybody ever done any microscopy? You've done it, so you do thin sections. And this stuff, we actually use a diamond knife, and the sections are so small that we can't even see them. So everything has to be done in a really high-powered dissecting scope. And the material, the stuff is embedded in plastic. And each piece is maybe not as big as a grain of pepper. So it's pretty sophisticated work. And let me just show you a few other things here. There's a Golgi, that's mitochondria, there's some ER. Those are mitochondria. Here's mitochondria from the cross-section. That's some ER, and there's some little ER there. And the big thing in the center is the nucleus. So what you're looking at is a cell that's doing a lot of stuff. It's got a lot of cytoplasm. It's got a lot of endoplasmic reticulum. It's got Golgis. So this cell is pumping out products. Okay? We're going to have a lot of RNA being made, transported to the ER, where proteins are going to be made. So if you look at this cell, you know there's a lot going on here. Okay? Now we're looking at the other end of what a cell can do. Where this is a cell filled with chloroplasts. So where would you find this cell? Probably in the leaf, maybe in a green stem. But we're going to the absolute opposite extremes here. Now we have a cell that is carrying out photosynthesis, and almost the entire amount of the space is what? The big vacuole. So remember, you've got a balloon inside a balloon. So that vacuole, the inner balloon, is filled up. The chloroplasts are pressed against the cell wall, and you'd want them to be pressed up against the cell wall, because what do?
They need light.
They need light, okay? And if I can back off, when you're looking at a tree, you're really looking at chloroplast. Most of the tree, most of the leaf is actually colorless. It's just got a bunch of chloroplast in it. And in fact, you're only looking at a small portion of the chloroplast. There just happens to be a ton of chloroplast in every leaf. The cell walls, for the most part, are thin, and light goes right through. Leaves, when they're not wilted, the leaves stand up to, of course, increase the surface area for light absorption. Okay? So this cell is active, but if I go back to this one, I would guess that this cell has a primary cell wall, and it's probably producing material for whatever. And I can say this is probably a leaf cell. Okay? Now, what do organelles do? If you go back to a bacteria, you've got basically just a bag of stuff. And there's a little tiny bit of organelle type structure. But basically, we have this compartmentalization. And so when we have compartmentalization, we can have different things in different places. Things are only not happening in the cytoplasm. What happens in a chloroplast? Photosynthesis. What happens in mitochondria? Anybody remember? You get a bunch of ATP produced. Okay? So these compartments have evolved over time to allow for stuff to happen in a certain area of the cell. Now, I'll tell you now that we're pretty sure mitochondria and chloroplasts came from the same thing. You know, they became, over time, they got into the cell and became part of the cell. You'll see, we'll talk about later, I'll tell you now, mitochondria and chloroplasts have their own DNA. They reproduce by division. Okay? And these things have really highly structured three-dimensional shapes. A lot of what you can get in this class is that you look at something, it's not flat. You have to figure out it's going in front and behind the plane. So a mitochondria can be sort of like a undulating type worm type thing. Chloroplasts are about 3 times as large as a mitochondria. Chloroplasts are easy to see in the light because they should be green. And these also talk to each other. So a chloroplast has its own DNA, makes a lot of its gene products, but it also needs stuff from the nucleus, as does the mitochondria. Okay? It gets pretty complex. Okay, I remember I've told you this I don't know how many times. I did this room. And chloroplast C is a little bit bigger than a mitochondria, and plasma membrane is extremely thin. You don't need to know these relative. You do not need to know the absolute size. You'd want to know that the vacuole is basically the largest organelle. And now we're back up, and now we've talked about plasmodesmata. Let's talk more about the structure of plasmodesmata and how they can look. So if we look at the top left panel, this is a micrograph. And you're seeing the cell wall here. Then you're seeing the absolute openings, and that these plasmodesmata actually have some structure to them. And you don't need to worry about the names of these things, other than the fact that they are open and that they can have ER that moves from one cell to the other cell. And we also find, that's not on this picture, that there's some degree of the ability of the cell to open and close this hole. So it might actually be able to close it down and open it up, close it down and open it up. So you got a hole through the cell wall that can be lined with membranes. Okay? And like I said, that's thought this is one mechanism by how in which viruses can spread through a plant. So just as an aside, you may see labs where they put shoots in culture and tissue culture and grow them really fast. And the idea is they can grow the shoots higher, faster than the viruses can move. So then you cut the top off, and now you have a piece of plant tissue that does not have any viruses. You're sort of making them grow real fast, and the viruses are going through the plasmodesmata, but the plasmodesmata are put down a little bit in like the zone of maturity. So then you can take the top off, cut the growing tip off, and grow it, and it may be virus-free. And that's one of the most common ways that people get virus-free plants. That's what Dr. Miller does, if you've ever seen his potatoes up on the 5th floor. Questions? Now, plants, just like animals, they have this very sophisticated transport system. Okay? And again, this is going back from what I said at the beginning, is that we would name these things part of all of the same system now, if we were to start naming them now. And we're going to go over each individual one, but let me just talk about them now as a group. And we have the nucleus, of course. And one thing that the nucleus and the chloroplast and the mitochondria have in common is that they are surrounded by 2 membranes. Remember I told you last time that a membrane is 2 halves? So the chloroplast, the mitochondria, and the Golgi have 2 complete membranes. They have 4 halves around them. Okay? And this is a pretty neat picture to try to give you sort of an idea of what it looks like in 3 dimensions. The endoplasmic reticulum is an extension of the outer membrane of the nuclear envelope. And what happens on ER? What's the number one thing that happens in rough ER? Anybody remember? Synthesis of what? Protein synthesis. Why is it called rough ER? What makes it rough? Bumpies? Anybody know what the bumps are, remember? Ribosomes. So if we knew what ribosomes were before we named it, we'd probably name it something else. But it's called rough ER, and the R, the rough, are the little dots, the ribosomes. And so you have material coming out of the nucleus. What comes out of the nucleus to make protein? mRNA. We're going to talk about that too. So it moves out, and then in the rough ER, it can then be made into proteins, and then you see some transition areas here. You see this, what they call a smooth ER, and then you have vesicles that go to the Golgi, and a lot of times in the Golgi, you have further processing of the proteins. Glucose units can be added to it. Carbohydrates can be added. Let's go back to the cell wall. When cell wall constituents are made, the proteins that synthesize the cell wall parts are made in the ER, and then as it moves through to the Golgi, then the galactoses, the rhamnoses, all those carbohydrates are added in the Golgi, and then the Golgi then can send vesicles out to the cell membrane, and it's the cartoon right here shows you the contents being dumped out of the cell. Now what's cool is we can go from here in the nucleus, so we got a pore here, all the way out, and that material has never been in the cytoplasm. It's always been surrounded by a membrane. Why is that important? Because if that stuff was in the cytoplasm, it would be chopped up by enzymes and that are in the cytoplasm. So those vesicles protect that special products, and we'll stick with the cell wall precursors that can go all the way from RNA in the nucleus to the cell wall and be dumped out, to the cell membrane and be dumped out of the cell membrane without ever being really in contact with the cytoplasm. Okay? So now we'll go back to this. Again, a little bit different. We go back to the smooth ER is usually considered the transition between the ER and the Golgi. And if you want to make a model of rough ER, take some saran wrap, put, I don't know, beans on it or something, glue little beans on it, and just randomly roll it up. And that's what an ER is going to look like. Just sheets after sheets of membrane. What is the purpose of all these sheets? These long sheets with the ribosomes on it. Increases the surface area for protein synthesis, okay? Smooth ER is where some other, like carbohydrates, are added, and if you want to build a model of smooth ER, grab some garden hoses up and twist them in all possible combinations. And that's what the smooth ER would look like.
You said that was a transition from what to the Golgi?
The smooth ER is sort of between the rough ER and the Golgi, yeah. Again, we call it smooth ER because it doesn't have ribosomes on it, okay? If we were to name it now, you know, the rough ER would be the zone of protein synthesis. The smooth ER would be the transition zone of whatever and the Golgi would be the area of glycolization or something like that, you know. That's where a lot of the carbohydrates are added and other things. So, here's a good picture of a nucleus. Let me go back. I wanted to say that, of course, we're talking about this thing. And here you see, what's this right here? That's a chloroplast. What are these things? Mitochondria. What's this big thing out here that you can't see? Probably the vacuole, okay? So what do we see in the nucleus? Well, we see the nuclear membrane here, and we see something called the nucleolus or nucleus, and that's about it. What you see in this one there. What's that? That's a really good one. What's that? That's a Golgi, yeah. And you can see the little tiny bits of ER there. And that's about it. So the nucleus has a very complex structure. This picture at the top is what you can do is if you freeze the tissue and you put it in a vacuum and you freeze it. And inside that vacuum is a very, very fine razor blade that you can literally move from outside. And it shatters through the membranes. So you freeze it, and what you see then is the top, the middle, and the bottom of the membranes. So think of a membrane being in a half. So you're going to shatter either over the top of it, through it, or below it. And you see this sort of these depressions, which turns out to be very, very complex nuclear pores. Okay? You don't need to worry about these structures here. What you do need to understand is they can be open or closed. And so the only way for material to get from the nucleoplasm, let's say it's RNA, is to go through these nuclear pores, these holes. So you can sort of see them here. And they're pretty complex. And there are people that just study these smaller proteins that really determine what goes in and out of the nucleus. You have the DNA in the nucleus that never comes out of the nucleus. Then you have the RNA, the transfer RNAs, and then the messenger RNAs, which copy the messenger RNA, which, as you know, copies the DNA and moves it out for the RNA to be used for protein synthesis, and mostly the rough ER. Okay? And you can open and close that. Now we're going to talk about the endoplasmic reticulum. So we go back to our standard slide, and we look at where we're seeing this. And where we're seeing the endoplasmic reticulum is always adjacent to the nucleus, because it's basically taking the material from the nucleus, the RNA, and turning it into protein. And here's just a micrograph of the two kinds of ER. Up close, you basically can start seeing the individual dots. So you don't see a good shot of the membrane here. And on this left is, of course, rough. And on the right, these tubules that you can see. You're here seeing a cross-section is the smooth ER. Okay? So usually we go from rough to smooth. So if we go back, we're going from the nucleus to the rough endoplasmic reticulum, then into the smooth endoplasmic reticulum. Okay? Now, these things have three-dimensional shapes, and here's a cartoon of the rough ER. And so what you've got are just these piles of membranes with the ribosomes on them. And there's all sorts of structure to this. And again, when you see a micrograph, you're just seeing that one layer, but they're going to go in and up. So just grab a bunch of saran wrap and just sort of roll it up, and that's what you've got. You know, this massive amounts of membrane, which increases the surface area in that very small cell to allow a lot of synthesis to occur. And if this works, let's see if it works. There we go, it is working. This is actually a film of the endoplasmic reticulum, more the smooth than the rough. So they were able to put a protein in, they were able to put precursors in here, and since this is where proteins are synthesized, the proteins, in this case it's green fluorescent protein, so you can actually see exactly where the protein is, okay? And again, this is more smooth ER than rough ER. So it forms this big network. But if you go back and look at this, you know, you think it's just this, well, it's just this little sort of static thing. No. It's a fairly large network that things can move around in. Okay? Then we want to talk about the Golgi. We've got a Golgi here, Golgi apparatus. And we see a lot of things associated with Golgi. Here's some mitochondria. Here's Golgi. Here's some nice Golgi. And if we see Golgi, if we see a lot of Golgi, we're probably looking at a plant cell that, let's say, is making a lot of cell wall material. Or maybe we're seeing proteins having carbohydrate added to them. And usually when you see this, you're also going to see mitochondria. Here's a mitochondria. Here's a mitochondria. And they're pretty complex. And what Dr. Mollenhauer did, here you see, this is the Golgi that he actually described. And what he did, he and a few other people, is, okay, you're cutting this section that's so small you can't see it. You have to look at it through a microscope. And so what he did was keep cutting section after section, doing serial sectioning, and then with wood making a model of what he was seeing. Okay? So what you're seeing on the right there, that's not a great picture, is an actual wooden model of what a Golgi looks like. So you've just got these membranes, and inside these membranes reactions are occurring. Most of the time the reactions are adding precursors that are going to be used in the cell wall. Can be other things can happen in there too. But you get an idea when you see these, you get this nice picture on the left, and you've all heard Golgi being described as pancakes, right? Like a stack of pancakes with blebs coming out. And you see that right, you see, well, I can see that, you know, but you just don't see 3 dimensions. We can now, in some instances, see 3 dimensions. And then the vacuole. Here's a good shot, not a great micrograph, but a good shot. Here's a fully active leaf cell with chloroplast in it. What's this big thing right here? Anybody know? That's the nucleus. The rest of these things are chloroplast. I put this in here because you can see it's just a large, it's the largest organelle. Some things that people don't think about is that this vacuole can store water. So the plant can actually, if needed, can use some of the water that's in the vacuole. It's a place where a lot of color is stored. So water-soluble pigments are, for the most part, in the vacuole. And it's a place where waste can be stored. There are a lot of times there are crystals in the vacuole, and those crystals could be minerals that could be reused. So you can put stuff in the vacuole, and you can take it back out if you need it. Okay? And of course, what the vacuole controls a lot is the water relations of a plant. So the vacuole, that inner balloon, is pushing up against the outer balloon, which is the plasma membrane, against the cell wall. Okay? So you've got water that's pushing and forming the cell shape. So this leaf would be standing up. This would not be from a dehydrated leaf. Now, the next two things we'll talk about are chloroplasts and mitochondria. And we'll be revisiting these when we talk about photosynthesis and respiration. Their structures are incredibly complex. If we look at the mitochondria, we have this thing where ATP is made. It has two membranes. It has an outer membrane and it has an inner membrane. And man, you're talking about being zoned out. I think I want to stop. I'm sorry. I've had 24 course hours in microscopy, so I could go on, you know. So anybody have any questions? You will have some micrographs to identify probably. You know, in my first course I had, on Friday you got a micrograph. The person that identified the most organelles got the highest grade. So every Friday we'd all get these micrographs, and we had, that's Golgi, that's Golgi, and you know, don't worry, I won't give you some that are really, really hard to view. But you know, I want to say I like doing this work. I'm still doing microscopy now, and I can tell you guys are so interested in it. So we'll pick up on Monday. Any questions? Anything? Go ahead, and if you're going to do the Google Plus, let me know if you're having any problems.